Welcome to lesson 3, video 2. We are talking about titrations in this video. And here we have a diagram of some of the equipment crudely drawn by myself, but nonetheless, it will get our point across. So here we have a burette. And again, this is fitted with a valve right here that you turn that's called a stopcock. There is a very small hole that drains down to a point, and you can control the amount of solution that's in the burette drop by drop if needed, but it does come out in a very small stream. But by turning the valve, you can open and close the burette, but you can also control how fast the liquid comes out. This starts up here at 0 and goes down to 50 milliliters. So it is a reverse scale. So if this were 50 up here and you drained it down to 10 or drained it down to 40, you'd have to subtract it to get your amount. So by having this as a reverse scale, you know that if it drains down to 10, 0 to 10 is 10. So it's able to be read. And we always are going to read this to 2 decimal places. And we always read at the bottom of the meniscus. So right here would be an example of the liquid, and it forms that U shape. So we always read it at the bottom, and it is read to 2 decimal place values. The last place value is estimated. So inside this burette is a standardized solution. Which means that it is of known molarity, and the volume used will be recorded. So we are going to know how much we have. We know its molarity, so we know the volume, we know the molarity, so we know how many moles are going to react. This standardized solution is prepared using a primary standard. So this is the burette. This right here is called the stopcock. And then down here, we have a flask. And in this flask is a solution of known volume or mass. So we know we can quantify this amount. So if we dissolved something in a quantity of liquid, we know the mass of what we dissolved, which is going to react with whatever we add from the burette. But unknown concentration. And it has an indicator in it. So the indicator is added in here. So as we add liquid into here, a reaction takes place and it uses up the known mass of reactant here. So we're going to have some steps to a titration. First, we are going to determine the concentration of the standard solution using titration with a primary standard. Our second step, we're going to do a second titration. So we're going to add the standard solution to the unknown until the endpoint is reached. And lastly, we're going to determine the unknown concentration using stoichiometry. So we will have a demonstration of how this works in a lab setting, and then we will be working on some problems.